Unfortunately, there's no possibility for the audience to clap, but I think they would do it if they had the opportunity. And let's move on to the next talk, which is from Pierre Morisse. Pierre, we can see you. I think we could hear you during the test.
Yep.
Next, yeah, we can see
share my screen.
Let's share your screen, and I will introduce it.
Should be okay, I guess.
Yes.
Perfect.
Excellent. So next talk is from Pierre Morisse, who is also at GenScale. And his talk is about LEVIATHAN, efficient discovery of large SVs by leveraging also long-read data. Go ahead.
Okay, so hello everyone, and thank you, Ryan, for the introduction. I'm Pierre Morisse. I'm currently postdoc at INRIA Rennes, and I'm going to present you LEVIATHAN, which is a new tool for discovering large structural variants using linked-read data. So to give a bit of context, structural variants, or SVs, represent variations in the structure of an organism's chromosome, and there exist many kinds of such variations, which include deletions, duplications, insertions, inversions, and translocations. And these variations usually range between 50 base pairs up to 3 megabase pairs. And they are especially interesting because many SVs are actually associated with genetic diseases. But the main problem is that SVs are usually much harder to detect than classical SNPs. A few words on linked reads also, although Anne gave pretty much a best introduction than what I will present here, but it works by partitioning and barcoding DNA molecules prior to classical short read sequencing. And as a result, the origins of the short reads can be inferred from the barcode, so we can determine which short reads originate from a common DNA molecule. And basically, linked reads give access to a high sequencing quality as well as to a long-range information, because the molecule lengths usually range between 10 to 50 kilobase pairs on average. Although, as Anne said, multiple molecules can share the same barcode, so this can, of course, complexify the analysis. So we have access to various sequencing technologies for producing linked reads data, the most well-known being 10x genomics. But we also have other technologies that emerged in the last 2 years, which are STLFR, haplotagging, and TELSEC. And in the context of this work, we're interested in detecting SVs using these linked reads. And this is actually something we can do using a barcode analysis. So for instance, if we realign our linked reads to a reference genome, if we have no SV, we should be able to see stuff like this with all the blue barcodes gathered together and all the orange barcodes gathered together. But if we have, for instance, an inversion on the green region right here, we will be able to see such abnormalities with blue and orange barcodes gathered together on the left and on the right of the green region. And basically, this will give us pretty strong indication that there is most probably an SV occurring here. So to give a few words on the state of the art, most methods work by first dividing the chromosomes into windows and trying to compare the barcode contents of these windows, because actually distant windows that share a higher number of barcodes than expected should be the sign of a SV. And if we take as example a heat map here, we can see that between this locus here and this locus here, we have quite a strong number of shared barcodes, as we can see here. And this is a pretty strong indication that we should have an SV appearing here. So to actually call SVs in linked reads, we have 9 tools which are available as of today, which are listed right here. But the problem is that most of these tools are designed for and overfitted on human data and are also no longer maintained. And as a result, applying these tools to non-model organisms is pretty difficult because they have quite a large number of pretty serious limitations, since some of them can be applied to fragmented assemblies, so assemblies that have more than 1,000 contigs, and some others require more than 15 days of runtime, even on short data sets. And as for the memory consumption, some of them also require sometimes up to 1 terabyte of RAM. So this is basically what motivated the development of LEVIATHAN. And it works as so. So it takes as input a BAM file, and the main novelty of LEVIATHAN will be a barcode indexing strategy. So we will build an index from the barcodes, And using this index, we will be able to compute the number of common barcodes between all possible region pairs. So we will have all our counts here, and from this count, we will compute the empirical distributions of the number of shared barcodes. And using this distribution, we will be able to retain region pairs that only share a sufficient number of barcodes. And we will further analyze these region pairs using classical short-read methods, so read pairs and split-read signatures, to determine the type and the breakpoints of the structural variants before outputting them in a VCF file. So the pre-processing step will be the barcode indexing step. So we will try to index the occurrence positions of each barcode. And if we give an example here, the index will basically be a map with the barcodes being the keys and the values being a list of occurrence positions. So for instance, here we have that this barcode appears on chromosome 0, position 100, chromosome 0, position 3500, etc., etc. And basically using this index, it will be pretty easy to match any barcode occurrence to the chromosome region, but also easy to identify region pairs that share a given barcode. So to do a bit of advertising, this is performed using LREZ, which is a toolkit and a C++ API that allows to manage linked reads data and that has lots of other functionalities. It was presented at poster number 8, so if you still have a bit of time after the talks, please go take a look. Anyway, back to Leviathan, the first step will be to generate the SV candidates list. So for now, we are only interested in identifying the regions where SV may occur, but not in their precise location and their type. So to start, we will divide the chromosomes into windows of a given number of base pairs, which is 1000 by default. And we will browse the index to process each barcode independently and iterate through the list of its occurrences. And so for each region pair we can build, we will increment the associated SV support of this region pair. As a result, we'll be able to compute the number of common barcodes between all region pairs without having to use a double iteration on the regions. So to give an example, if we take again our previous index, to compute the number of common barcodes between regions, we will start with this occurrence that we will match to the following occurrence. And if we choose regions of size 1000, we have that this occurrence here corresponds to this region, so between 0 and 999, and that this occurrence corresponds to this region, so between 3000 and 3999. And so this couple will basically increase the support of this region pair. And we will keep going as so exploring all possible region pairs until we are done with the first barcode. And we will keep going with other barcodes. So these two occurrences will increase the support right here, etc., until we reach the last barcode. So once again, these occurrences will increase the support here. These two will increase the support here. These two will increase the support here. And we will keep going until everything is processed. But as we can see, we have lots of regions that actually only share a single common barcode, and we will have to select valid SV candidates because, of course, further analyzing all these region pairs would be extremely time-consuming. So we will actually filter out region pairs that share an insufficient number of barcodes. And as I said, we will do this by using an empirical distribution analysis. And actually, we will study 3 different distributions. So one for close region pairs, one for moderately distant region pairs, and one for very distant region pairs. And we will define as a threshold above which we will consider SV as valid candidates, the 99th or the 95th percentile, whether we run Leviathan in fast or sensitive mode. The third step will then be to type the variants and compute the actual breakpoints. And to the same, we will retrieve the alignments in the 2 regions we previously identified for the SV candidates. So for instance, if we have a region here and here, we will extract all alignment for both these regions. And we will use classical short-read methods, such as read pair signatures. So for instance, in the case of a deletion, we will want to check if we have a greater distance than expected between the 2 mates of a given read. And we will also use split reads to try to see if we have splits to determine the actual breakpoints of our variants. The fourth and final step will then be to filter the candidates and output the SVs. And we will only consider a candidate as valid if its support is high enough, where the support is determined according to the number of barcodes, number of discordant paired reads, split reads, etc. And we also have an additional filtering procedure because the same SV can be represented by several different candidates which have close breakpoints but actual different types. And in this case, we will only report the candidates which had the highest cumulative support, so according to the number of barcodes, etc., etc. Finally, we will output the SVs in a VTF format, indicating, of course, our type, beginning and end position, the length of the SV, and all the support information, including the number of common barcodes, number of discordant paired reads, etc., and everything support related. So to give a few experimental results, we used both simulated and real data. So the simulated data, we're using LRSIM to simulate 10x genomics data on the first chromosome on the human genome and on a draft assembly of Heliconis pneumata butterfly with 17,000 contigs. And for the real data, we once again use real data from Heliconis pneumata because on this data, we are interested in finding large inversions that impact the wing color patterns of these butterflies, as we can see right there, but also the population biology. So if we first take a look at the results on simulated data, so on the human dataset, we can see that Valo2 required more than 2 days of computation, and it was killed after 2 days. That Grok SVs and linked SV both crashed after respectively 6 hours and 19 minutes. So we can only compare Leviathan to Neighbor and Lagrange. And we can see that the fast mode of Leviathan reached the highest precision compared to all other tools, but that its recall was also pretty good. So pretty comparable to both Neighbor and Lagrange. As for the sensitive mode of Leviathan, it reached the highest recall, but the precision was also pretty good, actually much better than both Neighbor and Lagrange. And if we look at the runtime, we can see that the fast mode of Leviathan was much faster than all other methods, and especially Lagrange. Although Lagrange also performs additional tasks such as alignment and SNP calling and so it's actually pretty normal that the runtime is a bit slower. And then if we look at the memory consumption, we can see that Leviathan was very efficient and especially much more efficient than Neighbor on this dataset. If we then switch to the Heliconis pneumata dataset, we can see that actually not much tool managed to run, where Valo2 crashed upon start, Lagrange was killed after 15 days of computation, and both Grok SVs and linked SV crashed after about 20 minutes of computation. So we can only compare Leviathan to Neighbor. And once again, we can see that the fast mode of Leviathan reached the highest precision, that the sensitive mode reached the highest recall, and that anyway, in both cases, Leviathan was much better than Neighbor. In terms of runtime, Leviathan Fast was once again faster than all other tools, and Leviathan Sensitive, however, was a tiny bit slower than Neighbor. And once again, in terms of memory, Leviathan was pretty efficient, and this time especially more efficient than linked SV. Then for the real Heliconis pneumata datasets, actually only Leviathan managed to run on these datasets because Lagrange couldn't run because the draft assembly had too many contigs. Both Grok SVs and Neighbor required more than 15 days of runtime. Linked SV required more than 1 terabyte of RAM, and once again, Valo2 crashed upon start. In comparison, Leviathan only required 2 hours of runtime and 18 gigs of RAM. It managed to report a total of 50,000 SVs, and especially it did manage to locate the 430 megabase per inversion we expected at its correct breakpoints, where the breakpoints were previously identified using SNPs, which were associated to strong sequence divergence between individuals. So to give a quick conclusion and a few stay-home messages, Leviathan is a new tool for calling SV using linked list data, which compares pretty well to the state-of-the-art, at least on simulated data, and which allows a reduced runtime and memory consumption, and is as so the only method able to apply to non-model organisms, as we could see in our experiments on real datasets. The specificities of Leviathan is the innovative strategy of barcode indexing. So once again, a bit of advertising, LRZ poster number 8. And this barcode indexing strategy allows to compute efficiently the number of barcodes between all possible pairs of regions. And in turn, this allows a quick determination of SV evidence. And this basically allows Leviathan to be very resource frugal, both in terms of time and memory consumption. The tool is available on GitHub and as a bioconduct module, so you can also flash the QR code right here to directly access to the GitHub page, and the preprint is also available on bioRxiv. So future work will focus on further validating LEVIATHAN with other real data, especially from the Genome in a Bottle project, to see how it behaves on other data. We also wish to apply it to other non-model organisms and other sequencing technologies, and especially the haplotagging technology. And we also wish to develop a local assembly procedure that would not only allow us to handle insertions SV, so detect insertions, but that could also help us better detect the breakpoints of the SV. So a few acknowledgements to conclude: to my postdoc supervisors Claire Lemaitre and Fabrice Leger, to the ANR Supergene grant for funding this work, and to Mathieu Joron and Paul J for the availability of the Heliconius new method datasets. And of course, thank you very much for your kind attention, and I will be very happy to take questions.
Thanks, Pierre. Any questions from the audience? You should also feel free to raise your hand if you want to ask a question instead of typing it. Yes. I will briefly continue on a question I asked to Anne in the previous talk. So main argument for using 10x seems to be cost compared to long reads. So do you have like examples of projects where it's clear that you have some large advantage in lower costs in 2020 or 2021?
Using 10x genomics, I'm not sure, since they also discontinued their technology, but I know that haplotagging is very cheap and it's also an open protocol, so it's very interesting. And it allows to do, I think, a very large number of sequencing in a single run, and it's very cheap. So I think it's very, very interesting in regards to long reads data. But I think pretty much, as Anne said, both can still coexist because we can still use both kinds of data to basically detect different kinds of SV. So I'm not sure we really have long reads versus linked reads, but we should rather try to kind of walk hand in hand and trying to use both data to get the most information we can.
Okay, thanks. Still nothing from the audience, so I will take the opportunity to rebound on this. So did you make a slide about haplotagging? Because it seems to be an interesting preprint from 2020 about sequencing lots of haplotypes from butterflies. Can you tell us more about it, please?
Actually, I have no slide for properly presenting it, and I didn't have time to properly read the paper yet, so it's on my to-do list, but I have pretty much lots of stuff to do, so I didn't have time to get to that point yet, so I unfortunately can't say much about how it works exactly.
It's okay. It still sounds like an interesting development.
But it sounds very, very promising, yes. So I guess it's a very, very interesting paper to take a look at.
A question from Véronique Bruno, who asked, thank you, what is the limitations of your method?
The main limitation would be if we have variants for which we can't determine a large number of barcodes between the two main regions of the variants, we will not be able to detect them. So this is something we actually discovered not so long ago. So we are currently trying to think about how we could try to identify these variants, because maybe that solely relying on the barcode evidence wouldn't be sufficient for detecting all variants.
Thanks. Well, let's give it a few seconds to see if anyone wants to ask one more question. Yes, Roland for, can LEVIATHAN detect all types of variants with the same precision?
From the experiments we did, it actually did manage to detect pretty much all kinds of variants with the same precision, yes. But as I said, we still don't have a procedure for detecting insertions variants, so we cannot detect them currently, but we are actively working on it, and we're hoping that what we are coming up with will work just as well as the other kind of variants we are able to detect.
Okay, sounds good. So, and Claire just gave a link to haplotagging, which is apparently now published in PNAS.
Oh, yeah, thank you, Claire.
Thanks, Claire. Yeah. So thanks again, Pierre. Interesting developments on linked reads from REN in this session. I will have to consider this technology closer for the future. And so thanks again, and let's move on to the last talk of the session.
Thank you.
So the last talk